Okay, good day everyone. So I test my mic first. Okay, so testing. Okay, my line is okay, mic is okay, camera okay, slide okay, everything is okay. Okay, so hi everyone. So good day everyone. So good evening everyone. So today we will have our last class for semester one lah. So today I think is the last 2 day for year 2023, right? So hopefully, luckily we have can I able to teach you guys some one syllabus in 3 months. Yeah, no mistaken, 3 months, right? So I already today is our last class. So all the chapter I already proceed. So just that the enzyme chapter I not finished yet. So Tuesday, right? Tuesday we already finished the enzyme reaction, right? Enzyme mechanism. So enzyme mechanical meter, maintain constant and linear plot. So all these I hope that everyone is can understand it. So today we will finish up the chapter 4 enzyme, which is cofactor, inhibitor, classification, and also enzyme technology. So first we go to the cofactor and inhibitor first. Okay, so let us see. Okay. Okay, so actually cofactor and inhibitor, these two compound substance actually is not enzyme. It is just help, either it's help enzyme or prevent enzymatic reaction. So cofactor is to help enzyme one. It is the non-protein substance, normally is non-protein substance that can take part in enzymatic reaction and are regenerated for further reaction. So normally this cofactor is to help the enzymatic reaction or enhance the enzymatic reaction. They are speed up the reaction. But another one we call as inhibitor. So inhibit, right? Inhibit meaning is prevent enzymatic reaction. That kind is also non-protein substance. It is also the non-protein substance. So these two cofactor and inhibitor are not enzyme. It just act as the precursor or alternative substance that can affect the enzymatic reaction. So we go to cofactor. So as we know that cofactor, we have three type of cofactor, right? Three class of cofactor: ion activator, coenzyme, and protective group. So what are they? So how they bind the reaction? So normally ion activator. So you see ion. So normally the example is mostly is metal ion lah. Metal ion. All of them are metal ion because it is inorganic ion. So the key point is attached temporarily to the enzyme, meaning that this ion activator only can attach temporarily only. It will not bind to the enzyme one. Bind to the enzyme for the long terms one. It just temporarily. If the reaction is completed, maybe this ion activator will be released out from the enzyme one. Just temporarily only. Just sekejap je. Sekejap je. Dia tak akan bind sangat lama punya. Kejap je dia akan hilang. Dia akan keluar lah. So this is the key point you need to know. Okay, so the function is to change the active site of the enzyme become more compatible with the substrate. So the metal ion is to ensure that the enzyme active site is fixed to the substrate. So this one is one example of induced fit model lah. If you know that induced fit model, the active site of the enzyme is not fixed to the substrate one. It need to change the shape first, baru then it can fix to the substrate. So this alteration is caused by one of example is ion activator. If have ion activator, metal ion bind to the enzyme, then it can change the shape. If not, the enzyme cannot. In certain situation lah. So the example of the ion activator is this. So all of them are metal ion. You will see here, inorganic ion lah. But mostly is metal ion lah. So calcium ion. So these are the example enzyme, and also the process that responsible to this ion. So you know that calcium ion is responsible to this thrombokinase. So this thrombokinase, the function is in blood clotting, right? You still remember this one as you learn in SPM. Zinc ion is responsible in DNA polymerase. So the DNA polymerase is responsible in DNA replication. This one you learn in Sem 3. So senior, you will know it, but junior, maybe you not know, you just memorize it. Ferrous ion is catalase. So normally this one is hydrogen peroxide degradation. So this one is toxin. Hydrogen peroxide degradation meaning it is a toxin degradation. Hydrogen peroxide is toxin to human one. It play the role lah. Magnesium ion is glucose 6 phosphate. So this one is glycolysis lah. You already learned it. Chloride ion is cytochrome oxidase. This one is play the role in ETC lah. And another one, manganese ion is play the role in arginase. This one is in the role of ornithine cycle. So for your Sem 1, maybe you can memorize calcium ion, you already learned it. Maybe magnesium ion, chloride ion, you also can learn it lah. And got other example, if you have other example, yeah, you can memorize it. There are many examples actually. This is just some common example in our biological reaction. Okay. Okay, another one is called enzyme. So, okay, so called enzyme and another one we call as protistic group. They are something like similar, just that the binding is different only. Coenzyme, the binding is loosely to the enzyme. It bind loosely, but for protistic group, the molecule is bind tightly. So what are their difference? If you refer to ion activator, the ion activator main difference is this inorganic ion. But for the coenzyme and protistic group, it is organic molecule one. And then organic molecule one. So if you refer to inorganic, memang metal ion only. And if you refer organic, either is coenzyme or protistic group. So what are their difference? The difference is this. Coenzyme is bind loosely, but protistic group is bind tightly. Ini depends dia. And then they also change the shape of the active site. So the function is same. Just that coenzyme bind loosely, protistic group bind tightly. So for protistic group, it will become the part of the enzyme known as holoenzyme lah. And then it will change from one form to another and back takes place in a single catalytic cycle. Okay, and another one is called coenzyme. So coenzyme normally is readily detached to act as carrier that transfer chemical group, atom, or electron from one substrate to another. And then it can exist in two or multiple form. So normally this coenzyme can exist in two multiple form. Either in oxidized form or reduced form. So actually coenzyme, most example is coenzyme A lah. You already learned in Krebs cycle, right? Acetyl CoA. How to form acetyl CoA because of coenzyme A. So actually coenzyme A is one type of coenzyme memang. And another one is ETC that you learn in coenzyme Q. Coenzyme Q. Ubiquinone, you know that. Coenzyme Q. So it is also one type of coenzyme. So normally this one coenzyme A. So this one is the structure of coenzyme A. So actually coenzyme A is very complex substrate. This one. So actually it is the main component is made up of vitamin B lah. Vitamin B. So actually the coenzyme normally your coenzyme is made up of vitamin B. Your vitamin B, you see here, your vitamin B, all of them are the precursor to form the coenzyme one. The origin of the coenzyme is vitamin B. Vitamin B. You see here all these many of them like NADH, FADH, coenzyme A, all of them are come from vitamin B. Okay, so this is the coenzyme. So protistic group, the example is Hem group. One of the example is Hem group. Hem group. So this one, right? Hemoglobin, we know that hemoglobin, hemoglobin contain Hem group, right? So ini Hem group lah. But you say that got inorganic, but with this one we refer to all these all organic molecule, not refer to only ferrous ion. If you refer to inorganic ion, that is only this ferrous ion that play the responsible. But for heme, it does not only ferrous ion. There are also all of the complex heme group that play the role. So you cannot say that this one is inorganic lah. Okay. Another one, yeah, biotin. Okay, another one is biotin. So this biotin normally play function in transport of carbon dioxide. This one you learn in gases exchange SEM 2. SEM 2, gases exchange, the transport of carbon dioxide. You learn about here, senior pernah dengar tak biotin. Ni paling famous lah, energy carrier. So all of the energy carrier, NAD, FAD, NADP, ATP, all of them actually are under protestic group. All of them is under protestic group. So FAD, FNN, not only FAD, FNN also all are under the protestic group. So normally there are many example, semua ni example lah. So all of them, you see here, all of these are all the example. Okay, so this one is vitamin B. So vitamin B is very important lah. It is the origin from the coenzyme and also protestic group. Both coenzyme and protestic group is origin is come from vitamin B. So you see here, thiamine, vitamin B1, the coenzyme is thiamine biophosphate lah. And another one, riboflavin, FAD lah. And another one, nicotinic acid, NAD+, pantothenic acid, coenzyme A. Biotin is biotin, lysine, adduct, and then B12, deoxyadenosine cobalamin. So all this lah. You just pick the famous one lah, like riboflavin, thiamine, and another one this nicotinic acid. Pick this one and also pantothenic acid. Okay. So we know that vitamin B is act as the coenzyme. So only vitamin B. The other vitamin A, vitamin C, K, D, E, that one does not involve. Only vitamin B. So normally these are all the vitamin, vitamin B5, B2, B3, B6, and then another this all of them are responsible that act as the coenzyme. And then this one, vitamin K, vitamin A, E, and also vitamin D, vitamin D, all of them actually is not, they are vitamin, but they not play the role in coenzyme. So please don't confuse that. Only vitamin B play the role in coenzyme only. Vitamin A, D, E, K, no, they not play. Okay, so you need to remember it lah. So in objective question maybe come out lah. Vitamin, if you say that vitamin A act as the coenzyme, yeah, no, only vitamin B. And then in here also, that's all for coenzyme. So any questions? So actually coenzyme is very easy topic lah. Okay, no. Okay, next we go to inhibitor. Okay, so inhibitor, if you know the names, what is mean by inhibition? Inhibit. Inhibit, we know that inhibit is prevent, right? Prevent something happen. This substance can prevent something happen. We know as inhibitor, inhibit, right? So normally inhibitor in enzyme, meaning that this is the substance, any substance that can prevent the enzyme reaction, decrease the rate of enzymatic reaction or prevent enzymatic reaction. This one we call as inhibitor. Any substance, not tak kisah dia organic, inorganic or other lah, apa-apa je. If this substance can inhibit the reaction, decrease the reaction, prevent the enzymatic reaction, all of them we known as inhibitor. Okay, so this inhibitor is very important in syllabus. It also very hot topic lah. So there are in our syllabus there are two main types of inhibitor: competitive inhibitor and also non-competitive inhibitor. So got other one is we call as uncompetitive inhibitor, but this one is not in STPM syllabus. NPM tak mention, so you don't need to learn it. You just learn these two type only. Learn these two type only. Sorry. Two type only. So apa itu competitive and non-competitive? So inhibitor actually is the substance that can bind to the enzyme to prevent reaction. Dia juga bind to the enzyme, but this one is not enhance the reaction but prevent the reaction, meaning it prevent the substrate to bind to it. Kalau cofactor dia bind dekat enzyme, but it enhance the reaction. But this inhibitor, it bind dekat enzyme, bind with the enzyme, but it prevent any reaction. No reaction. It prevent the substrate cannot bind to the enzyme. So no reaction. So how it play the role? So competitive inhibitor, meaning that it is the substance inhibitor that have the similar shape to the active site of the enzyme. So dia macam abang, macam twin untuk substrate. But this one bukan substrate, because when it bind to the enzyme, no reaction one, no reaction. It does not have the reaction at all. Does not same as the substrate. If substrate bind to it, it has the reaction. But this, if competitive inhibitor bind to it, although it bind to the active site, although it has a similar shape, but this one is act as inhibition. So no reaction. So when it bind to it, it will cause the substrate cannot bind. Yang original substrate punya tak boleh bind lah. So yang ni awak boleh cakap in general, awak boleh cakap boyfriend kan, boyfriend girlfriend, a pair of how to say satu boyfriend girlfriend lah. Tapi lepas tu boyfriend tu enzyme, girlfriend tu substrate. Tapi boyfriend tu suddenly dia cari orang wanita lain lah. Dia cari wanita lain, kawan dengan dia. So meaning that wanita lain is act as the inhibitor lah. Inhibitor like that lah. I can say like that lah. Example. So the competitive inhibitor is, the main point is it have the similar shape to the active site of the enzyme. This is the key point. Another one non-competitive inhibitor is the inhibitor will bind to the what we call as intrinsic site. Intrinsic site. INTIRNSIC. Intrinsic site. Or another you can say other than active site. If it is other than active site or allostatic site, allosteric site, if it is bind to the allosteric site, meaning is totally, if you see here, this diagram more easy. So if the enzyme is bind to the other part of the active site, so normally this one we call as allosteric site. So when bind to it, it will change the enzyme active site shape. The enzyme active site shape will be changed, totally changed at all. Dia akan berubah punya. Kalau dia berubah, so when the shape of the active site is changed, meaning that it is now not complementary to the substrate now already. So it does not have the same shape to the substrate, the original substrate. So this substrate cannot bind to this enzyme anymore. But competitive inhibitor, you see here, the inhibitor have the same shape, so it directly bind to the active site. So this one, because it already blocked, so the substrate cannot bind to the active site lah. Kerana dia dah ada orang dekat sini lah. So ini substrate memang nak pergi ke tempat lain lah. But for ni, walaupun dia ada kosong, but now this active site is already changed the shape. It does not have the similar shape to the substrate. So although it have the empty space, but the substrate now cannot bind to it. Dia tak boleh bind lah. So this substrate cannot bind to it. So the substrate will also cannot bind to the enzyme. So these are two main difference. In the enzymatic reaction, two main difference. But there are also some difference. Later we will see it. So what are other difference? You need to refer to the Mitchell-Manton graph and also linear bird plot. Linear bird plot. So for those who are still not understand apa ini graph ni, apa itu linear bird plot, please make your revision. I will really teach you last Tuesday. If you not attend, itu nasihat apa lah. Kenapa kalau tak nak attend? Okay. Anyway. Okay, so normally if you see here, this Mitchell-Manton graph, this black line is normal enzymatic reaction. Ini normal punya. If you see here, when competitive inhibitor is present in the reaction, you found that your graph will be like this. Your graph will be like this. Like this. What are their difference? Apa difference perbezaan dengan ni? Based on this graph, Vmax, half Vmax, Km, and also substrate concentration. You will found that the Km value will be decreased. Dia akan increase the Km value, right? You will increase the Km value. Right. Kalau macam ni lah, ini Km ni sepatutnya dia dekat sini for original one. But for now, your enzymatic reaction, you see here your Km here now at here for red line. Because this one is half max lah. If you refer to half max, keluar sini, ini original Km. Kalau tak ada inhibitor. But for now, because of you have the competitive inhibitor, you found that your Km at here, meaning you increase Km value. So, as I mentioned, when your Km value is increasing, is high, meaning that your enzyme and your enzyme is not have the high affinity to the substrate. Meaning the enzyme dia tak suka substrate. Dia tak akan bind dekat substrate itu. Kenapa? You will see here, you will know that competitive inhibitor dia ada similar same active site with the enzyme. Kalau dia bind memang substrate tu dia tak boleh bind lah. So kalau boleh bind, the affinity between enzyme and substrate totally decrease. Memang akan kurang punya. Kerana enzyme dah kurang. Your concentration of enzyme that can be worked for the substrate is already decreasing because already got inhibitor that block the space already. So meaning that your your affinity between enzyme and substrate is already decreased. So you will get Km is decreased. But your Vmax is maintained. You maintain your Vmax. Your Vmax is still maintaining. Why? Because this competitive inhibitor, you only bind to the active site. You not change the shape. So if your substrate concentration is increasing, you can overcome the competitive inhibitor reaction. Meaning that when your substrate concentration is always increasing, your this competitive inhibitor will be released from the enzyme. Dia akan release punya based on equilibrium lah. So meaning that competitive inhibitor it is the reversible reaction. Dia reversible punya. Kerana inhibitor tu memang dia boleh lari daripada enzyme punya. Kerana dia hanya simulation, it just block it only. Dia hanya halang je. Kalau terlalu banyak orang, memang gate tu akan buka. So kalau dia keluar dari enzyme, memang ada reaction punya. So the Vmax will remain. So this is the competitive inhibitor. So when you compare the competitive inhibitor between the normal enzyme, the Vmax value, your maximum enzymatic reaction is same, but just that your Km value will be increased. You will increase your Km value because at your initial time, your substrate and enzyme is infinity one. Your affinity of substrate and enzyme is very low. Okay, but when you refer to the non-competitive inhibitor, you will found that the main difference is the Vmax lah. You found that your Vmax is decreasing. Awak boleh tengok. Your Vmax should be normal enzyme is at here, very high. Like maybe is 10 lah, 10. But for now, it decrease to 4 like that. So meaning that your Vmax is decreasing, but your Km value is remain the same. So meaning that your Km value still remain at here. You remain your Km value. Why? Because non-competitive inhibitor is bind to the allosteric site, which is not the active site. They are bind dekat sini. So it already changed the shape. So when the enzyme is already changed the shape, meaning this enzyme cannot use anymore. Tak boleh guna. Kerana walaupun this non-competitive inhibitor is released out from the enzyme, but the shape of the enzyme is already fixed already. Dia tak boleh ubah. Dia tak boleh change back to the original one. Tak boleh lah. Dia dah directly change. So this one non-competitive is is irreversible reaction. Dia tak boleh reverse punya. Hanya satu direction punya. When non-competing inhibitor bind, then it will cannot change already. The enzyme, the active site shape will that cannot change back to the original one. So this one will cause the enzyme concentration, your enzyme, your working enzyme in the solution is decreased, totally decreased at all. Compared to competitive. Competitive, initial they are decreased. Tapi when your substrate concentration is already increased, that enzyme still can work. That enzyme affected enzyme still can work because the active site still fixed with the substrate, so it still can work. So your working concentration, your working enzyme in the reaction actually still same with the normal enzyme one. Sama je. If your normal, for example if your normal enzyme in reaction, your normal enzyme have 100 enzyme, ada 100 enzyme dekat satu dekat solution itu. So in your normal enzyme memang 100 enzyme tu boleh buat reaction. But for your competitive inhibitor, let's say lah, if your competitive ada 20 competitive inhibitor, so dia bind dekat enzyme, maksudnya your initial time you only have 80 enzyme work only. Dalam mula-mula hanya 80 enzyme yang bekerja je. Yang ada 20 kerana dia ada block tak boleh kerja. But when your enzyme concentrate, your substrate concentration is increased, so your competitive inhibitor will be released out from the enzyme. So the other 20 affected enzyme will be work anymore. Once the inhibitor is released from active site, then the enzyme can work. So actually the competitive inhibitor, the solution still have 100 enzyme that can be worked. Just that initial tak boleh kerja. Tapi kalau substrate dia tinggi-tinggi, memang tak boleh kerja lah. But for non-competitive inhibitor, when there are 20 types of non-competitive inhibitor in the solution, memang 20 enzyme to 20 enzyme cannot work anymore. Although the non-competitive inhibitor released from the allosteric site, but the enzyme confirm cannot work anymore because it is irreversible. The enzyme already changed the shape. The active site of the enzyme is totally changed. So it cannot back to the original one. So meaning that this working enzyme only have 80. You only in maximum hanya 80 je. So memang kalau compare to normal enzyme, your maximum enzymatic reaction will totally decrease. So your Vmax will totally decrease. But your Km is still remain the same. So as logically lah, kerana dia affect tu memang affect lah. So maksudnya the substrate and enzyme, the working still the same. The affinity between substrate and enzyme still the same one. Kerana yang affect memang affect, dia tak boleh ubah buat apa-apa lah. Tapi so substrate hanya boleh cari yang boleh kerja je. Yang ni kompetitif, dia akan cari dua-dua. Either dia tak boleh kerja atau boleh kerja kerana dia tak tahu. Because they found that fixed shape right, just that dia ada halangan dekat sana. Tapi yang ni memang awak pergi cari dia yang affected punya substrate cari yang affected punya memang tak boleh bind lah. So dia akan automatic cari yang boleh kerja punya. So the Km value will still remain the same. So these are the main differences lah between competitive and non-competitive. So this one is when you use the Mitchell-Manton graph. Another one you use the linear line variable plot. Line variable plot you will see like this. So actually this one normally it will come out want you to calculate lah. Dia akan bagi equation. Maybe they will come up with equation. What kind of equation you still remember, right? 1 over velocity enzymatic, we add 1 over enzymatic rate versus 1 over substrate concentration. So and then you know that your s-intercept is negative 1 km, right? And then your y-intercept is 1 over Vmax, and then your slope is km over Vmax. So this one is very basic one lah. You need to know it lah. Memang kena awak tahu lah. So apa macam mana dia datang? Please refer back that I teach you last Tuesday. So actually what I need to mention is totally same lah. Yang red color is your normal reaction. So if you see here, if the competitive inhibitor, your Vmax will maintain the same, but your Km value, your Km value is affected lah. Dia akan cari lebih besar sikit lah, -1/Km. So you see here, dia become more positive lah. This one is negative feature. You see here, the original one is more smaller. Yang ni lebih bigger sikit. So meaning dia dah increase lah, Km value is already increasing. So, okay. Another one, you see non-competitive inhibitor. So, you see here, dia lebih tinggi kan. The slope is more increased, right? The slope. You see, you refer to there, these three, if have the inhibition, the slope is more increasing. Your gradient is increased. But for non-competitive inhibitor, it is have the highest gradient. And then you found that your Vmax now is more higher value, but this one is 1/, enzymatic value. So if very high, if the value is very high, meaning that your Vmax is actually decreased. You are decreasing your Vmax, but your Km value still remain the same. So okay, so ini graph, these two graph you need to memang kena remember punya, and also based on these two graph, actually you already know what are their differences. So if exam, when you come out you difference between competitive inhibitor and non-competitive inhibitor, you know how to do it. And also in exam, maybe dia keluar graph ni want you to find out the Vmax, Km, or even gradient, maybe will come out one. Saja maybe dia nak kamu keyword equation, find the equation, maybe dia nak cari, normally would want you to find intercept lah. And then based on this intercept, determine which this is the competitive or non-competitive. So based on you find the Vmax, and then you compare to the normal one. So do you think this is competitive or non-competitive? Like that lah. Okay, so I think that's all for cofactor and inhibitor. So any question? Oh, no question. So okay. So now I will proceed to the enzyme classification and also enzyme technology, the last two topic for this chapter lah. Okay, so if no question, I will proceed. Okay. So classification of enzyme essentially is very easy topic lah. Memorize lah, tak ada pilihan lain. Memorize lah. So in IUB, this classification is based on this IUB society, International Union Biochemistry. So this society already classified the enzyme into 6 classification. But for now, the latest one have 7 classification. Ada satu lagi we known as translocase. But in your syllabus, MPM, kerana MPM tu, dia ambil syllabus dekat 2012. So, that time, ya only have 6 type. But nowadays, already have 7 type. But anyway, based on the STPM, we just based on the MPM syllabus lah. So, MPM syllabus cakap ada 6. So, ikut je lah 6. So, okay. So, these are the all 6 lah. Lyase, transferase, ligase, oxidoreductase, hydrolase, and also isomerase. So you need to know this all, and also what are their role. Lyase, dia buat apa? Transferase, dia buat apa? So first we go to oxidoreductase. So actually this is very general lah. You see here oxidoreductase, oxy and reduct. So already clear. Meaning this one is play the role in oxidation and or reduction. Oxidation and reduction process one. So any reaction, enzymatic reaction involve oxidation and reduction, the enzyme is called as oxidoreductase. So you know that right? Oxidation, apa itu oxidation reduction? So oxygen, you transfer oxygen, release hydrogen, accept hydrogen, release electron or accept electron, ini semua kan, all of this is involved in oxidation and reduction, right? So one of the these are the 2 examples. So the example, this one I take for, this one is glycolysis, this one is Krebs cycle lah. You see here, this one is about oxidation, and then this one is about the reduction. Yang ni ada reduction kan. So okay, so example lah. So you can refer to it if you So if exam, kalau exam dia keluar and then it want you to give example, you just refer to your chapter 5, chapter 6. Any chapter 5, chapter 6. Any chapter 5, chapter 6 that involve oxidation and reduction, you can write it. Any reaction. These are the two examples, but actually got more. You just refer to it. Ada example, right? Okay, another one is hydrolase. So, dia pun sangat senang. Hydro. Meaning, dia include water. Hydro. But hydrolysis, hydration, all of that is include water. So, the reaction that involve hydrolysis reaction meaning dia ada tambah water punya with one add water molecule one. So, memang hydrolysis reaction is the enzyme is known as hydrolase. So this is the example. When Krebs cycle one, fumarate to malate, the enzyme is fumarase, right? So ada water, right? Hydrolysis reaction, hydrolyze. So okay, so you can use the example. Use this example. Senang pun. Okay. Another one is isomerase. So ini pun senang. Iso, as we say, isomerism, iso, meaning this one is isomerization. So if the reaction that include isomerization, meaning that isomerize is meaning what? Same chemical formula, chemical formula, your chemical formula is same, but your structural formula is different only. So one of the example is glycolysis lah. Your glucose 6-phosphate is isomerized to form fructose 6-phosphate. Inilah one of the example. And another one is also glycolysis, your intermediate one. Your intermediate one, I already forget, you can refer to it. So this one is another one of the example. So isomerase. Another type is lyase. Okay, lyase, this one is come from lysis. So if you lysis, lyase. So lysis apa maksud? Meaning dia break chemical bond. Kan? Break chemical bond. But this chemical bond break other than redox reaction at hydrolysis. So other than redox reaction and hydrolysis, so normally redox reaction and hydrolysis, they are still can be breaking down your chemical bond. But because of that, they already mentioned oxidation, reduction, or hydrolysis. So that kind is specific to them only lah. So other than this one, and got chemical bond breakdown, this one called as lyase. So what are the example are these? In glycolysis, you know that in glycolysis at the step 4 or step 5, step 4 or step 5, your fructose 1,6 biphosphate will be lysed, will be split out into these two molecules, right? Dihydroxy acetophosphate and also glyceraldehyde 3-phosphate. And then these two molecule, ini saya cakap lah, ini two molecule is isomerized, right? Dia boleh isomerize to each other. So these two molecule also isomerization. And then this whole process is called a lyase. So the enzyme involved is aldolase. So this aldolase is classified under lyase. So you need to know. If you want to give example, you can use this example. Another one is transferase. Okay, so this one is involved when you transfer a functional group to another from one substrate to an acceptor molecule, meaning you transfer one functional group to another substrate. So one of the example is this lah. The first reaction of the glycolysis glucose to glucose phosphate. So you already transfer, right? This enzyme transfer the phosphate group from ATP to this glucose molecule, right? So this what we call as phosphorylation. So ini example transferase. So semua senang kan? So actually you just memorize glycolysis, then all the enzyme you already know. Apa semua enzyme you dah tahu lah. So kalau exam keluar ni memang if you memorize glycolysis, then you automatically memorize also the enzyme classification. But for this ligase, but for this ligase is not in glycolysis and Krebs cycle no this reaction. But for this one, it is the symmetry one, catalyze the formation of new bond between the two substrate molecule by using energy from ATP. So this one is not involved in your glycolysis and Krebs cycle, no lah, because this one it will form new bond one, form the new bond. The substrate molecule between two substrate, this one involve two substrate molecule. Two substrate molecule, when there are two substrate molecule need to bond together, this one we call as ligase. So this one will not involve glycolysis and Krebs cycle, because glycolysis Krebs cycle mainly is only have one substrate, right? But for this ligase, it will must be involve two substrate. So the example actually this one is in SEM 3. SEM 3 one. SEM 3 you will learn it. So the reaction is amino acid with tRNA. You will, with the enzyme we call as amino acid tRNA synthetase, you will form the activated tRNA we known as amino acid tRNA. So this reaction actually is in translation process. You learn in SEM 3. So for senior, who repeat, you know it lah, by two. But for junior, you just memorize it lah. You don't need to know it. I just give you clue. It is under the translation topic, sentence 3 you learn. Okay, so that's all of the 6 class of the enzyme. So actually it's very easy because except the ligase, other oxidoreductase, hydrolase, isomerase, transferase, and also lyase. These 5 enzymes you can take the example from the glycolysis or Krebs cycle. Actually, you take from glycolysis is already enough lah. All of 5 of them already have in the glycolysis one. But for the ligase, example you can use this lah. This is the example lah. Dia tak ada include glycolysis Krebs cycle one. Okay, so I think this one is all about enzyme classification. So enzyme classification, got question? No question. Okay, very good. Okay, now we go to the enzyme technology. Okay, this one is about in application of enzyme lah. Under the application of enzyme, this one for those who want to learn whatever if you want to further study biotechnology, one of the branch enzyme technology is one of the branch if you want to go to biotechnology, and also bioinformatics. You want to go to bioinformatics or biotechnology or pure biology also can. You will learn one of the branch is you want to make a research in the enzyme. So you want to make a research in the enzyme, then you will learn about this lah. Okay. So normally enzyme immobilization. So what are the meaning of enzyme immobilization? So this is the meaning, the definition. This is the technical process which that we fix our enzyme in the solid substrate, creating a heterogeneous immobilized system. Meaning that when in industry, in our industry reaction, because nowadays, because of green technology, enzyme nowadays is become a biocatalyst, very useful catalyst already. Because in old generation, the industry is using chemical. They're using the chemical to act as the catalyst in the reaction. But nowadays, because the chemical is very toxic to the environment, it's not eco-friendly. So nowadays they use enzyme. But enzyme is very expensive. If you know that enzyme actually is very expensive. If you want to buy the enzyme, it's very expensive. So if you want to increase the enzymatic rate reaction, you need to use this technology we know as enzyme immobilization. Meaning that we will got one reaction, your enzyme is located at that part only. Hanya dekat satu tempat je. Dia tak lari punya. Because if you go to the if you know fix tak akan not fixed to the solid support in certain type of location, your enzyme is freely in the solution, but dia boleh bergerak ke mana-mana punya. And then after that, when the enzyme already formed the product, we know that enzyme can be reused. It can be reused one. But for now, after the enzymatic reaction, your enzyme, your product is remain at here. So we only one product. Kami tak buat enzyme because enzyme kami nak reuse again. Kami nak recycle, reuse. But for now, your solution is now contain enzyme and and your product. We only want product. Kami nak product keluar je. So, kalau kawan nak buat product, awak kena buat reaction lagi lah. Nak buat apa? Filtration, chromatography. You need to further process. So, waste money lah. So how to fix it? We fix to the enzyme. So when the solution inside the substrate, inside the solution, your enzyme is fixed at there. And then after the reaction, you can directly remove the enzyme easily. So meaning that after the enzymatic reaction, you can directly take out the enzyme, and then that solution only contain the product that you want. So you save your money save your money to extraction purification process. So that is why that we want enzyme immobilization. So this one of the advantages advantages. So actually inilah apa yang saya cakap tadi tu inilah. So kenapa kami nak guna immobilize enzyme? So enhance productivity and stabilization of enzyme. You need to stable your enzyme. We know that enzyme actually is very unstable compound. Because enzyme is made up of protein. You know that protein is very unstable one. It is very easily to be denatured at the extreme environment. So use this technology, you use the immobilized enzyme, you can stabilize it. Maksudnya awak boleh stand in the high temperature environment. You can stand in the sudden change in pH value. Boleh dekat extreme environment lah. Another one, ini saya cakap lah. If your end product is not contaminated by enzyme resulting no end product will be not be contaminated by enzyme resulting no purification. Ini yang saya cakap lah. Because in one solution you only want end product, kami tak mahu enzyme punya. So if you not do immobilized enzyme, memang awak kena buat purification. But for now, if you using your immobilized enzyme, you easily take out enzyme or you easily remove the product. Your enzymes remain at that place. You remain at that tanker lah, dekat tanker tu, and then you release out all the product, and then your enzyme is remain at there, and then after that you can reuse this enzyme every time lah. Another one, reuse anytime. So compared to the freely enzyme, if you use immobilized enzyme, you can reuse many times, allow continual process, banyak kali. Compared to the freely, you can reuse, but freely maybe if your environment is sudden change or you have the purification apa-apa je, your enzyme maybe can be denatured or destroyed. But when you do this, you will fix them together so that the enzyme is more stable, so you can reuse the enzyme in many times lah. And then yang ni saya dah cakap lah, not easily to be denatured. And also, it can occur in non-aqueous solution medium, where the solid support preserve the enzyme constituent and make them stronger. Normally in your lab, in general theory, freely enzyme only can involve the reaction in aqueous medium only. Non-aqueous medium cannot. Like non-aqueous medium cannot have the reaction one. But if you use the immobilized enzyme, non-aqueous medium, you can proceed to it. Okay. And then there are another two. So normally, can alter from homogeneous to heterogeneous form after the enzymatic binding, which assists in separating the enzyme producing product with high purity. So meaning that you will not only occur, this enzyme will only work in the homogeneous form. Homogeneous form meaning in the solution. You also can work in heterogeneous one, in solid form. Awak boleh work dekat solid form. Meaning your enzyme is solid, and then you pour the substrate, and then they have the reaction, and then directly you extract your product out. So this one we known as heterogeneous form. Kerana ada solid dekat sana lah, kerana you immobilized enzyme is solid form. And then reaction can be terminated rapidly by removing the enzyme solution. You can directly terminate it. Compared to freely, you want to stop the reaction, very difficult lah. Tak boleh lah. Kerana kamu tak boleh directly remove all the enzyme in very short. This one you can terminated directly because you directly remove enzyme or directly remove product. Yeah, that's all. You can directly, very in short time. So these are all the and another is cost effectively lah. Another one lah yang paling senang punya cost effectively lah. Saving cost lah. Itu. Okay, so in our syllabus, so you refer to this. In our syllabus in MPM, syllabus dia bagi you only learn about adsorption, covalent binding, and also entrapment. Yang ada ni, awak tak perlu belajar. Ini bukan syllabus awak. Ini actually got banyak. Yang ni lah, semua banyak punya. Adsorption, covalent binding, and entrapment. Encapsulation, cross-linking, affinity binding, bonding, ionic binding. Actually, dia sama je dekat sini. Just different lah. So, in our syllabus, just refer this, focus these three only lah. Okay, so adsorption. So actually adsorption. So normally this one is enzyme will be weakly adsorbed to an insoluble particle such as hydrogen bond. So these are the linkage. Maksudnya your enzyme you see here, this is your enzyme and then this is your carrier. Normally is solid particle lah. Solid particle, solid particle, this solid particle. This enzyme just weakly bind only. Dia bind tapi very weak je. So normally this bond so you will see hydrogen bond, short linkage ionic bond. Although it has ionic bond, but this ionic bond is very weak ionic bond. It is not a strong ionic bond. Or even hydrophobic interaction only. Okay, and then you will see this diagram, you will see here, this one is original one. So normally if you want to enhance the adsorption, you can add a surface modifier lah. When you add the surface modifier to the carrier, then you have the strong binding compared to no surface modifier lah. Okay, so these are all the explanation. So normally I want you to know that your material carrier normally is include resin, alumina, carbon lah. Ini tiga yang paling famous punya. So normally this method is very cheap and very easiest method lah compared to other one. It is very easy to perform and it is very cheap only. Because you just only buy this only. Because you only buy this and then you pour enzyme, you buy this material and then you directly pour enzyme, then that's all. But it is not very accurate lah because it only weak adsorption. Weak adsorption, so it is very easily to be released from the enzyme. The enzyme is very easily released from the material one, the carrier material. So normally this one is not very recommended in industry lah. Normally is conducted in research lah. And research small scale one, yeah, you can use it. But in industry normally they will not recommended. Because industry is large scale, you hasilkan banyak. So kalau awak ada enzyme dia release out, dia akan wasting money lah. Because you know that enzyme is very expensive. It is very expensive one. Okay, another one is we call as entrapment. Okay, so entrapment, you know that what we go to trap, right? Trap, terperangkap. Meaning kami terperangkap, we trap the enzyme in one solid. Macam ni lah. Ini solid matrix, lepas tu ada banyak enzyme dekat sini. Semua enzyme dia macam masuk penjara. Masuk penjara lah. Ini penjara lah. Ini penjara. Yang ni matrix ni memang penjara. Lepas tu enzyme ini satu kotak-kotak ini memang bilik dia, bilik untuk suspek bilik untuk bilik penjara lah. So the enzyme akan masuk satu bilik-bilik ni lah. So normally enzyme is entrapped with an insoluble particle prepared for organic polymer through covalent or non-covalent bond. Okay, so these are the explanation. So normally this So this one is very famous technique. There no very famous technique. So the advantage is the enzyme cannot escape. Yeah, it cannot escape. Compared to adsorption is easily escape one. But for this, kerana dia dah terperangkap dia tak boleh keluar lah. Memang tak boleh keluar. So, 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 uh, so normally your enzyme cannot escape, but your substrate and product can escape. So meaning that when you use this, when the substrate akan masuk ke sini, mereka substrate and product, substrate tu boleh diffuse kepada solid ni. Dia boleh masuk, tapi enzyme tak boleh keluar. And substrate boleh masuk, lepas tu ada reaction, lepas tu form product, the product boleh keluar. Maksudnya substrate ni macam keluarga lah. Keluarga untuk yang suspek lah. Yang ini semua ini penjenayah substrate untuk keluarga untuk penjenayah. Dia nak pergi penjara, lihat dia. So mereka boleh masuk. Masuk. Lepas tu lihat. Okay, they form a product lah. So mereka akan keluar lah. Say bye-bye. Mereka keluar. So substrate and product can easily pass through and remove. But enzyme tak boleh. Enzyme boleh hanya dekat sana je. Macam penjara lah. Itu penjenayah memang tak boleh keluar. Dia hanya boleh dekat penjara je. Yang matrix ni macam penjara polis lah. Dia tak tangkap dia, tak boleh keluar. Okay. So normally the advantages is high loading capacity, low fabrication cost, mechanical stability, and also lower mass transfer. So normally this one is very recommended in industry lah. But yeah, when you recommended, it's not easily to carry out lah. Another one is covalent binding. Yeah, if you refer to it, covalent bind. So memang enzyme is covalent bonded to the cable, to the support material. So ini memang ada satu strong covalent bond at here. Compared to adsorption, although it contain covalent bond, but that covalent bond is very big lah. Sangat big. Easily to be break down one. But for this one is more stronger, so it is not very easily break down. So normally is enzyme are connected with the support material by a very high stable strong covalent bond. So these are the example of the material support silica, polyamide, agarose, porous glass. This one. Okay. So these are the adaptation. So normally the advantages is to producing durable enzyme, obtaining a sufficient recovery of the enzyme. And then increase the stereo specificity of the enzyme, and then low probability of enzyme leakage. So these are the advantages there. Okay, so I think that's all for enzyme immobilization. So any question? No question. Okay.